There we go, now it's recording. Okay. So for announcements, first thing to tell you is I'm starting to feel sick. And I was like, I woke up a little sick. By the time I got here, I felt a lot worse. So I'd say that for two reasons. Yeah, one, keep your distance. Two, if you wanted to visit me during office hours today, I would recommend doing it online. I'll be in my office, but I would recommend doing it online and not putting it in my tiny little closet of an office because I'm sick. And the other thing to think about is possibly, hopefully not, but there's a possibility I'll be teaching from home on Wednesday if I'm sick. So make sure you guys check your emails before you come all the way to campus. You guys, it may take you to drive all the way here, then may not be here. Any questions about my sickness? All right, today I think is the day I'll re-release the exams so you guys can retake it. And remember, the retake, you don't have to do it. It's open book. It's not timed. You just go in there and take your time. Take all week if you want. 2 weeks, I don't care. But just make sure you let me know when you're done so I can adjust your grade. And if you get a lower grade, then that won't matter either because we'll just keep your original higher grade. So you have nothing to lose. Plus, your final exam is going to be based off of your old exams, right? I'll take questions from your old exams to put on your final exam. So it wouldn't hurt to take a look at it again. And then, like someone's already asked, if you've done the retake or don't intend to, whatever, and you would just like feedback and meet with me, I can tell you exactly what's the right answer, and why this one's wrong, why this one's right, then you get that 5% boost to your grade. So that goes for any assignment. Oh. So any questions about past exam? No? Okay. What else to remind you with? Lab will be in person this week, so make sure you bring your computers to be here at noon on Wednesday. Some of you don't have to be there because you've already done a lab and an emails you had said you don't have to go there. So unless you hear otherwise from me, be here at noon on Wednesday. But feel free to work ahead. There's a possibility you might not have to go. Any questions about lab? Okay. Well, I'm going to start attendance because it is 8 o'clock. That being said, first word for attendance will be sickness in honor of my failing health. Anyway, if I don't call your name, let me know. That means you're not here. Carr, Smith. Carr, let's see, he's here. No, yep, he's here. 1 second. All right, Newton, Faulkner, Stanley, Whitaker, Fisher, Sims. That's it. So everybody's accountable, correct? All right, let's get started. Turn the slide off so you can see the screen. We're jumping back into chapter 9. What we're looking at here, come on, is, let me get you. Anyway, this is where we left off. We're talking about chromosomes and homologous, or the homologous chromosomes and alleles. So this is a good time to remind you that alleles are different versions of genes, right? So again, the oversimplified version of what we have for the eye color, right? We say you either have the allele for brown eyes or you have the allele for blue eyes, right? It's the same gene that's located on the same location on all of our chromosomes, but you might have different versions of it, and that's what this is saying. So remember, homologous chromosomes carry the same genes in the same location of the gene, right? So whatever gene P is here, you know, it's going to be always on that part of the chromosome for everybody, every human. So any questions about that? All right, let's move forward and actually get into new information. There we go. Let's talk about Mendel's law of independent assortment. So we already talked about independent assortment in the last chapter, so we're going to talk about it again. Obviously, in this case, it'll be more in the context of of heredity in genetics as opposed to mitosis and meiosis, or excuse me, meiosis. Let's talk about it. Before we do, I need to introduce you to a new term that you need to know, which is a dihybrid cross. And before I talk to you about a dihybrid cross, I'll remind you what I said a monohybrid cross is, because we talked about that on Friday, so it's been a while. A monohybrid cross is when you're crossing plants as you would, purebred plants, and you're looking at one feature, right? So maybe the flower color or the pea color, something like that. You're only looking at one feature. A dihybrid cross, then, is when you're looking at something with two features, right? It's the mating of parental varieties differing in two characters. So not just one, but we're looking at two, and that makes things a little bit more complicated. So this next bullet point with these ifs, it's not the way things are, but if the genes for these two characters were inherited together, which they're usually not, but if that were the case, then you write this down because this is not the case. If that were the case, then the F1 generation, so remember it's the P generation and they make the F1 generation, and then they make the F2 generation, right? So if the genes for those two characters were inherited together, then the F1 would produce only the same two kinds of gametes they received from their parents. And the F2 would show a 3-to-1 phenotypic ratio. But again, you don't need to write that down because that's not the case. Genes are not inherited together. Sometimes they are, but we'll talk about that later, right? They're completely different. They're on different parts of the chromosome. Sometimes they're on different chromosomes themselves. Remember, like for humans, we have 23 pairs of chromosomes. So you might have a gene for one thing on chromosome number 7 and a gene for something else on chromosome number 20, right? So this is not what happens. That is what would happen basically if it was a monohybrid cross. But since they do not do it like that, since they sort independently, what actually happens is that the F1 produced 4 genotypes, right? Not just 2, because we're no longer looking at just one gene, right? We're looking at 2 genes, so 4 different alleles, and they produce them equally. Therefore, the F2 will have 9 genotypes, producing 4 phenotypes in a ratio of 9 to 3 to 3 to 1. And that's the thing I'm going to circle for you. Because I know it's a little bit complicated, and I don't usually like to teach you just tricks on how to pass a test. But when you see that ratio, 9 to 3 to 3 to 1, you should think dihybrid cross. Just to remind you, the monohybrid cross, I said, would be a 3 to 1 phenotypic ratio. So if you see a ratio that only has 2 digits, you should think of a monohybrid cross. When you see a ratio that has 4 digits, you should think of a dihybrid cross. Any questions on that before I show you like a visualization of all this information? Okay. Here's what we're saying. This is the first if I gave you on the left-hand side, but it doesn't happen this way. So the genes for the color of the pea, right, and the shape of the pea, those are inherited separately. If they were inherited together, you would get this though, right? You would get this 3 to 1 ratio. But because we're talking about separate genes, you get all kinds of possibilities, right? So big R, big Y, big R, or little y. The F1 generation, again, there's only one option, right? Because one parent's bringing nothing but dominant alleles, the other one's bringing nothing but recessive alleles. So no matter what, just like with the monohybrid cross, that F1 generation is going to be heterozygous. However, when you cross those, then you get all kinds of options, right? So this one's completely homozygous dominant, right, for both alleles. This one, actually, I don't even want to get into that much detail. I don't want to confuse you guys. Basically, again, since you guys are 100 level, this is 100 level course, you guys are biology majors. The important thing here is you see this 9 to 3 to 3 to 1 ratio, and actually tell you, okay, this is the dihybrid cross. We're no longer looking at just one character, we're looking at 2. And in this particular example, again, the 2 characters we're looking at are pea color and pea shape, right? Either green or yellow is the color, or wrinkly or smooth. Of course, you don't need to memorize that. This is just an example. And in my opinion, the most important thing about independent assortment, which is what we're talking about, is what I told you in the last chapter, which is that when all the chromosomes line up during meiosis, it's not like all the mom chromosomes are on one side and all the dad chromosomes on the other. They don't do it like that. They just line up independent of each other. And to me, that's more important for you guys than what I just taught you. But I teach out of the book, so there it is. The dihybrid cross does support the hypothesis that each pair of alleles segregates independently of each other. So all that important stuff I just reminded you of, where all the moms and the dad chromosomes don't necessarily line up on the same side. That's why this is important, right? Because basically, we're just showing you that what I told you is in fact true. And if you do dihybrid crosses, it supports that, right? Another way of saying that, the inheritance of one character has no effect on the inheritance of another. So think about that, and I'm just making stuff up again, right? If you have the gene for bad breath on chromosome number 7, then you have the gene for stinky feet on chromosome number 10, those things have nothing to do with each other, right? Because they're on different chromosomes. And remember, you have homologous chromosomes, one from your mom, one from your dad. So when you make sperm or egg, we don't know which allele you're getting, right? It's not like you're going to give them all the mom alleles or all the dad alleles. We don't know because they line up independent of each other. Yeah. So again, that is the law of independent assortment. Any questions about this? All right, the next word for attendance will be, let's just say feet, since we talked about that, stinky feet. The feet is the next word for attendance. Whoops. There you go. Read it too. Anyway, your book uses Labrador retrievers as an example. So here we're kind of looking at a dihybrid cross again because we're looking at two particular characters with these dogs. One is the color of their fur, and the other thing we're looking at is if they can see or not. So being black is a dominant trait. So black is dominant to brown. Also being able to see is dominant, right? Being blind is recessive. So we looked at these. Let's talk about this. This is the first time I've shown you this. This dog, right, is black and can see. We know that, right? It's a dog, like, hey puppy. We know it can see. We can obviously see that it's black. So we know its phenotype. Its phenotype for both characters is dominant, right? So because of that, we don't know the genotype. We know it's big B something and big N something, right? But that second one could be a big B or a little b. It doesn't matter because that's a dominant trait. As long as you have one of the big alleles, then you're going to have the dominant phenotype. Same with the N. We don't know if this dog's big N, or big N, little n. All we know is that it has at least one big N because it has that dominant phenotype. So any questions about that? All right. So really quickly, then it should be pretty quick. Again, this dog's black, so we know it has the dominant phenotype for the color. So we know it's big B something, right? Could be big B, could be big B, little b. We don't know. All we know is that it has the dominant phenotype. However, this dog is blind. And we know that blind is a recessive phenotype. Therefore, we do know that thing's genotype for the site. We know it's little n. It has to be. To have the recessive trait, you have to have homozygous recessive genotype. Same with this dog, sort of, is brown. We can see that it's brown, so we know that it has the recessive phenotype for the color. Therefore, it has to be little b, little b. We know that. Also, it can see. So that is the dominant phenotype. So again, we don't know that genotype. All we know is it's big N something. It could be big N, little n, we don't know. And then obviously this last one right here, it's black and blind. So it has both of the recessive phenotypes. So we know its genotype for both. It's homozygous recessive. So all that's pretty much a review except these blanks. I wanted to make sure you understand what those blanks mean when you see them. That means we don't know the full genotype. Any questions? All right, so let's say you were to mate two heterozygote Labradors, right? So they're heterozygous for the coat and heterozygous for vision. This is what you would expect if you were to like just do that over and over and have them have a bunch of puppies. You would expect about 9 to 3 to 3 to 1 ratio, right? So whether we're talking about peas or dogs or humans or whatever, then that is the case. And again, that's just an example. I'm not going to ask you any questions about Labrador retrievers on the exam. The most important thing here for the exam is, again, you see that 9 to 3 to 3 to 1 ratio, or a ratio that has 4 digits, which is think dihybrid cross, because we're looking at 2 characters. Then, of course, this other new information is the blank spaces. You need to know what that means. So, are there any other questions about these dogs? All right, we're going to talk about them one more time. because we're going to talk about using a test cross to determine an unknown genotype. There'll probably be at least one question about this on the exam. I don't think it's complicated, but some people do. You guys ever heard of a test cross? Probably not, that's okay. Test cross is the mating between an individual of a dominant phenotype, right? So we don't know the genotype, right? Just like I was talking about with the black dog that could see. We know it was big N something, big B something, but we didn't know exactly what its genotype is because it's dominant. So you mate that thing with an individual that is homozygous recessive. Because obviously if it's homozygous recessive, right, we know it's genotype, we know it's homozygous recessive. But in that case, like how many people know their dog's genotypes, right? That's not everybody does. But if they have a recessive phenotype, then we do know the genotype. You have to be homozygous recessive to show the recessive phenotype. So that's what a test cross is. The next slide I'm going to show you will kind of show a test cross in action. Like we have this dog, it's black, you can see, so we don't know what its full genotype is, but we can mate it with other dogs and help us figure it out. All right. So like I said, we have a black dog that can see. It has two dominant phenotypes, so we don't know its genotype. Oh, here we go, sorry. In this case, we're only looking at one of the traits too. We're making it simple again. We're no longer talking about a dihybrid cross, we're talking about a monohybrid cross. So anyway, this one's black, right? Dominant phenotype, we know it's a big B something. We don't know what the other thing is yet. We know this thing's brown, therefore it has to be homozygous recessive. When we cross them, then we can see what the puppies look like, all right? So if this dog right here was big B, then this is what we would expect, right? We would expect every single one of the puppies to have the black phenotype. And that's dumb. I don't know why your book did it like this, because what they did was like a lazy version of a monohybrid cross, or of a Punnett square. So I'll go ahead and do the full Punnett square. The results will be the same, but you guys have been used to seeing a full Punnett square, not this. So anyway, if this dog was big B, right, when it makes sperm, those are going to segregate. So big B. Of course, this dog over here, we already know it's little b. So put that on in here. So you can see when you do this and bring all these together, 100% of them are going to be big B, little b. Does that make sense? I don't want to have to draw all those in, but you can see big B and little b come together. 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 So again, we're just trying to figure out what this dog's genotype is. So if we made it with this brown dog and 100% of the puppies are black, then we'll know, well, it'll support our hypothesis that that dog is big B. Does that make sense? All right, so the other possibility, again, would be on this side. If that dog was heterozygous, you would do a Punnett square just like I did over here, and just like they kind of did here, then again did a half version. Anyway, so when you mate the dogs, if there was brown and black ones, then you would know that this dog is heterozygous, right? If you have any brown puppies, you would know that that dog is heterozygous. Does that make sense? All right, the one thing worth noting is this is all probability, it's all statistics, right? So there's a possibility if this dog was homozygous dominant, right? There is let me see, I'm gonna put this. No. If this dog was heterozygous, like we see here, and you mated it with this homozygous recessive dog, like you said, this is what we would expect, half black, half brown. However, it's possible that they could just be black, right? It's all a flip of the coin. So even though this is what we would expect, we still might have all black puppies, in which case we would probably misdiagnose it and say, oh, it's a homozygous dominant. Anyway, are there any questions about test cross? Okay, really quickly then, let's talk about the rules of probability. Speaking of which, genetic crosses obey the rules of probability. It's the same that apply when tossing coins, rolling dice, drawing cards, all that stuff, right? Tossing coins is the best analogy when it comes to genes because, again, we have two alleles, right? When you make a sperm or egg for any given gene, you're either giving it the mom version or the dad version, right? Or you've already made your sperm and egg and look at it that way. So if you look at any given sperm or egg in your body, it's a 50% chance whatever gene is that you're looking for either came from your mom, came from your dad, right? And that's what that shows right there. So again, this female is heterozygous. So if we were to look at all the eggs in her body and looking for this particular B gene, whatever it is, we were to pick just one of those eggs out of all of them, there's a 50% chance we'd get one that had the big B, there's a 50% chance we'd get one with the small b. That's all that says. But then, and this is where it gets a little bit more complicated if you don't know it, that part's kind of easy to understand, right? 50% chance of us picking out a mom gene or a dad gene. But then let's talk about the fact that then we're taking that one sperm and that one egg and putting them together. That's where the rules of probability get a little bit more complicated, because then you have to multiply to figure out what the chances are. So again, if this heterozygous female mates with this heterozygous male, what are the chances of their one baby being, I don't know, homozygous dominant? And the chances are 1/4, because it's half a chance, 50% chance that you're going to have a dominant egg, 50% chance that you're going to have a dominant sperm, and then put those things together, the chances of those two coming together, 1/2 times 1/2, and that's 1/4. So you might get a question like that on the exam, where I give you all the information and then you just have to do the math. I might say, what are the chances, and this will make a little bit more sense when we start talking about some diseases later on, but I might say, what are the chances of this normal mother having a baby with this father that has this certain disease? What are the chances that the babies will have the disease? And then I can make it even more complicated and say, what are the chances that it's going to be a girl with that disease? Because what are the chances of you having a boy versus a girl? You guys should know this one. Like, all right, who does not have a kid in here? Okay, I think you were the first to raise your hand. So when you have your kid, if you ever have one, what are the chances that your first kid is going to be a boy? Exactly, 50%, right? It really is a toss-up. It's equal chance, 50-50. Therefore, if I were to say, what are the chances? If there's some disease and it's like, 50% chance that you would have the disease, and then a 50% chance that it would be a girl, so you have to multiply that. So again, 1/2 times 1/2, the chance would be 1/4, or 25%. But that'll make more sense when we actually talk about those diseases. Right now, it's all just a hypothetical conversation. So any questions about this particular slide? All right, the next word for attendance will be coin. So we talk about flipping coins. All right, family pedigrees. To me, this is where it gets a little bit more interesting. This is where we start talking about things that might actually, I don't want to say benefit you, but might affect your everyday life a little bit more. Hopefully not, but this would be very useful if there's a certain disease that might run in your family, or if you're mating with somebody who has a certain disease that runs in their family. So your book points out that Mendel's principles, all these things we've been talking about for these pea plants, they also apply to the inheritance of many human traits. So I know we've been talking about peas, and you're probably like, who cares about peas? I don't even like peas. I don't. So why is that important? But again, it's because the rules are the same, right? So the probability, all that stuff's the same. So we're going to show you some examples. You should be familiar with this from the lab, some of it anyway. Widow's peak, right? This lady's got widow's peak, this one doesn't. This lady's got freckles, this one doesn't. This one has free hanging lobes, this one doesn't, right? So these are all dominant traits up top, recessive traits in the bottom. You guys are all familiar with these from the lab, so that's nothing new. Here's something new for you. So make sure you know this word for the exam. Someone's going to mess it up. It's easy to mess up. So the dominant trait does not imply that it's normal or more common. And we proved that in the lab, right? There was a lot of things that were more students had the recessive than they did the dominant, right? Sometimes that happens. So when we say, what is the one you see, whatever particular character you're talking about, whether it's widow's peak or freckles or color eyes, all that stuff we talk about, the word is wild type. Wild type is the one that we see the most in nature. Or I might even just say see the most, right? Because in a sense, you might say, we're looking at this class, right? Look at the experiments we did last week. So yeah, that's what the wild type is. And I asked this during lab last week, but how many of you have freckles? Just two, right? So that's a good example, right? Look, freckles, that's a dominant trait. Yet only 2 people in this classroom have freckles. So it's just a good example. Just because it's dominant does not mean it's more likely to be seen. Any questions about what wild type traits are? All right, that being said, let's talk about human genetics again. How can human genetics be studied? This is different than pea plants, right? Because with the pea plants, somebody said, all right, I'm gonna make this one with this one, I'm gonna study their offspring, and I'm gonna make those offspring mate with themselves, and I'm gonna study their offspring, right? You can't do that with humans. It's not ethical to say, all right, you and you, mate, I'm gonna study your kids. I'm gonna make your kids have kids with each other, right? You can't do that. So how do we do it with humans? Well, that's where the pedigree comes in, right? Geneticists analyze results of matings that are already happened, right? So we don't force people to mate, we just look at what's already happened. We assemble that information into something called a family tree, also called a pedigree, and this can be used to determine if a trait is sex-linked. I don't know if your book said that or if I made a mistake. I don't like that bullet point. I mean, it can be used to determine if a trait is sex-linked. They can also be used for a lot of stuff, so I don't know why I included that bullet point. Anyway, the next slide I'm going to show you shows a pedigree that happens to be tracing the incidence of free versus attached earlobes, which is not a sex-linked thing, but we know from lab it's either dominant or recessive, right? And from that last slide I just showed you. And because of Mendel's laws, we can deduce some genotypes, and that's what we're going to do very quickly. We're going to deduce some genotypes. And keep in mind, you might have something like this on the exam. So what do we have here? We have a pedigree, right? So, 3 generations? We got some kids here, their parents, their siblings, grandparents, all that. We can see that. We also have phenotypes, right? So we know that if it's purple or blue, whatever color you might, what color is that? Purple? Okay, not that it matters. But anyway, if it's colored, that means they have the dominant phenotype, which is the recessive phenotype, which is the attached earlobes, right? So if it's white, we know they have the dominant phenotype. So that's what we have as phenotypes, right? We don't have genotypes. But if we use what we know, we can figure some of this out. So to have the recessive phenotype, what must your genotype be? Yes, homozygous, specifically homozygous recessive. So we know all of these are little e, little e. They have to be, otherwise they wouldn't have the recessive phenotype. So we know that. That's the first thing we know from what we've learned. The next thing we can figure out, based on what we learned, is at least half of these other genotypes, we know that they are at least big E something, right? So like when we were talking about the dogs, we know there's at least one big E. Otherwise, they wouldn't have the dominant phenotype. Does that make sense? You guys with me so far? It's painstaking. I wish I could just draw on the screen. Anyway, so now we're halfway there. Now we can really start deducing and figuring out some other genotypes. For example, let's see. Let's look at this one. All right, this one's easy. So we know that one, little e. We know this one's little e, right? So we know this one, actually, does anybody want to guess? That's a big E something. So does anybody want to guess what the other one is? Yeah, little e, right? It has to be little e because one of its parents is homozygous recessive. So the only possible thing that they could have gotten from their parent, that one, is a little e. Has to be. Anyway, that's how we do it. I would spend more time going over this and figuring these all out, but it would take a while. Maybe I'll make a video and do that separately and post it if you guys want it. You guys can watch it or not if you want to. And maybe I'll also post something like this for independent work, where I'm like, hey, you figure this out. Yeah, and like I said, maybe it'll be on the exam, but it is a little time consuming. So if it's on the exam, It wouldn't be this version, obviously. Second, if I do give it to you, it'll probably be extra credits. So anyway, that is important, relatively important, like I said earlier, for your everyday lives, not for earlobes, right? Because who cares about earlobes? But there could be a certain disease, like I said, where like, oh, that runs in your family. Well, we need to figure out what the chances are of us having a child with that disease. And again, we'll talk about that actually next. So any questions about pedigrees? All right. Like I said, next thing we're going to talk about are human disorders controlled by a single gene. Here we are. We're still talking about single gene things here, right? Keeping it nice and simple. But now it's going to be a little bit more relevant to you. We're not talking about dogs or earlobes or freckles or pea plants, right? We're talking about diseases, real life diseases. These are examples that are real life, not like the fake stuff I said, where stinky feet or smelly breath, whatever. These are real things. Some disorders are known to be dominant or recessive, right? Depends on the disorder. And obviously, they're going to follow the same rules that we've learned earlier. So to have the dominant disorder, i.e. phenotype, you only need the one dominant allele. To have a recessive disorder, just like any other recessive phenotype, you would have to have both, right? You have to be homozygous recessive genotype. Here's some examples. You could study this, but don't memorize it. This is not a health class, so I'm not going to ask you, what are the major symptoms of cystic fibrosis? Or here's some major symptoms. What disease is that, right? I'm not going to do that. It's for your own information. I'm not going to ask you about it. But this is a good list. Here we have some recessive disorders. So to remind you, to have this disorder, you would have to be homozygous recessive, right? Because that disorder, that word might as well be phenotype. It is a phenotype. And then here's some dominant disorders. And we're going to talk about some of these specifically, especially dwarfism is a really interesting one, in my opinion. You're going to see why when we get there. Are there any questions about that table? All right. This is also a good thing to look into for independent work if you want. If any of these seem interesting to you, just look them up and learn more about them. Like how common are any of these? Or what are the other, right? This is a pretty Cliff Notes version, right? So what are the other symptoms of cystic fibrosis, for example? Anyway, first thing we're talking about is recessive disorders. Most disorders are recessive. Excuse me. Most human disorders are recessive. Any guesses why that might be? Why are there not more dominant disorders or more people with dominant disorders? After all, you only need one of the alleles to make it happen. You don't have to answer yet. Just think about it. Let it marinate, because later when we talk about dominant disorders, then we'll bring that back up. So when we're talking about recessive disorder, I have to point out that individuals that have a recessive allele but appear normal are called carriers. So you need to know what a carrier is. So to be clear, and I'm just going to use a letter, let's just say it's D, right? An individual that has a recessive allele but appears normal. AKA, this person would be heterozygous. That's just another way of saying they're heterozygous. It's a recessive disorder, so they're fine, right? Because they don't have two copies of that recessive allele, they only have one, so they're just fine. But they are called a carrier. And that goes back to the pedigree thing that I was talking to you about earlier. Those are the kind of things you might concern yourself with when it comes time for you to have children. I know I made sure my wife had a clean bill of health with her family. No, I'm just kidding. I didn't do that. But it is something you would consider, right? Does this run in your family? Are you a carrier? Especially if you are a carrier for a certain disease, you might really, really look into it then. It's like, hey, I'm a carrier. If you're a carrier, then that means, you'll see the math, but there's a 25% chance that our kid's going to have it. Anyway, any questions about what a carrier is? If I were you, if you were taking notes, you could just shorthand and put like carrier equals heterozygous, right? They have one version of the allele, but they don't have both, so they don't have the disorder. There we go. For example, with the D, if we have two carriers mating, like I just said, what are the chances of them having a kid that has the disease, in this case, being deaf? The chances would be 1/4, right? 25%. And you will get a question like this on the exam. And like I said, to make it more difficult, I might say, a carrier for hearing mates with a carrier, or with somebody else who's a carrier for hearing, or with the deafness allele, what are the chances of them having a deaf girl? 25% times, sorry, somebody trying to come in. 25% times 50%, right? So 1/4 times 1/2, so the chances would be 1/8. Let me show you guys that, sorry. But we know this is 1/4, it's 1/4 of chance of having a deaf child. We know it's 1/2 chance of having a boy or a girl, right? So when you multiply those things together, 1/2 times 1/4 equals 1/8. And this is it's not a math class, so I will keep it simple like that. And I'll probably just keep the fractions for something like that. Because does anybody know off the top of their head what percentage of it is 1/8? No, me either, right? So we'll just keep it simple. Anyway, any questions about this? All right, let's talk quickly about cystic fibrosis, even though I'm not going to test you on it. Your book mentions it, so will I. It is the most common lethal genetic disease in the United States, which is probably why your book chose to talk about this one as opposed to all the other ones. It is caused by a recessive allele, and about 1 in 31 Americans carries that recessive allele. So there you go. Now you know. Not that I'm going to ask you. That's another good thing for independent work. Again, you can look into it some more. What are more things you can learn about cystic fibrosis? Or, this is an interesting statistic, 1 in 31 Americans. What about worldwide? What are the chances of just any human having it? Or if you want to zoom into your favorite country, what are the chances of somebody in Italy having it? Anyway, any questions about cystic fibrosis? All right. Next thing we're going to talk about are dominant disorders. So we're done talking about recessive disorders. Let's talk about dominant disorders. Remember, you could almost swap the word disorder for phenotype because the rules are the same. To have a dominant disorder would be like just having a dominant phenotype, which means you only need one copy of the allele. You can be heterozygous or homozygous dominant. It doesn't matter. If you have that one copy, you're going to have it. The achondroplasia is an interesting version of a dominant disorder because, in this case, if you were homozygous dominant, it actually kills you. You're not, like, before you're even born. So there are no humans that have achondroplasia. Excuse me, there are no humans that are homozygous dominant for this because they die before they're born, which is an interesting thing, I think. So yeah, normally when you're talking about a dominant disorder, like I just said, you could be homozygous dominant or you could be heterozygous. Doesn't matter, right? As long as you have that one dominant allele, you have the disorder. But again, this is a different situation because the big A will actually be lethal in this one. Anyway, so a person that has achondroplasia has a 50% chance of passing that condition on to their children. And I'll show you the math on the next slide. But are there any questions so far about achondroplasia? You don't need to memorize any of this. If there's any questions about achondroplasia on the exam, it's going to be another one of those. What are the chances of this person mating with this person and the offspring being this or that, right? It'll be one of those questions. And if it is achondroplasia, all this information is going to be there to remind you. Homozygous dominant is deadly. It's a dominant disorder, right? So all that, I'll remind you of all that. It's not like you just have to memorize everything about achondroplasia. Like, what happens if you made a dwarf and a non-dwarf, right? It won't be like that. Here we go. Here's some examples. So we have somebody without achondroplasia, which obviously would be homozygous recessive, mating with somebody who has it, which obviously would be heterozygous, right? Because if they were homozygous dominant, they'd be dead. So you just do a Punnett square like we've already done. And when this person makes sperm, 100% of them are going to be little d, right? So no matter which one you look at, you're going to get the little d allele. When this dwarf makes eggs, right? Half of them are going to be big D, half of them are going to be little d. You put those chances together. Big D, little d is an outcome. Big D, little d, again, another possible outcome. Little d, a possible outcome. And little d, a possible outcome. So like I said on the last slide, in this case, there's a 50% chance if a dwarf and a non-dwarf mate, 50% chance that their child will be a dwarf. Oops, I circled the wrong thing. What I said is still true, but I circled the wrong thing. It's the dwarf, the heterozygotes up there. Anyway, any questions about that? Okay. What this didn't show you, and you could just do it on your own if you want, but what were the chances of, let's see, if you had two dwarfs mate, right? You could do the Punnett square on that and figure out what are the possible outcomes of that. All right. Like I said earlier, recessive alleles are more common. I asked you why. I said you didn't have to answer. I said let it marinate. But let's talk about it. Dominant lethal disorders are much less common than lethal recessive disorders. And that should make sense. Because if it's dominant, if it's lethal, it'll kill you. So, you know, when you're dead, it's a lot less likely that you're going to pass along that gene because you're dead. A good exception to that would be Huntington's disease, because a lot of times people don't even know they have it until about middle age. So there's a higher probability that these people will actually pass along the gene before they even knew they had it. But that's why Huntington's disease is one of the more common ones. Because obviously, again, if it kills you, you can't mate, therefore you're not going to pass along those genes. But again, this one doesn't kill you right away. You can look it up. I'm not going to ask you any questions about Huntington's disease. It's just a good example because, again, it doesn't kill you right away. So it has had a possibility, that chance of getting passed down from generation to generation. Any questions about Huntington's disease or dominant lethal disorders? Okay, we're going to skip this next part like we always do, the process of science. As always, I always recommend reading it and let me know if you have questions. There might be questions about it on the study guide. There will not be questions about it on the exam. Just skip through, especially if you like dogs, right? If you're interested in dogs. There we go. Let's talk about genetic testing. Speaking of all this, right? Because I keep saying, you know, if you're a carrier and you're making somebody else who's a carrier, this is important stuff, real life stuff for you. This isn't like photosynthesis where you might be like, oh, I don't care about that. I don't photosynthesize, but it's a possibility that you might have to deal with this kind of stuff one day. What is genetic testing? Well, they are tests that detect disease-causing alleles. Most of them are performed during pregnancy if the parents are aware of increased risk of genetic disease. So again, if you know something runs in your family, you might want to get it checked, right? First, you could check yourself, right? And then just do the math and be like, all right, what's the probability of us passing this along to our kids? And then after you had the kid, you could either, or once you've fertilized, you know, you can test. You can test the embryo. And this is really something you could write about with that. It's really controversial right now. So for example, how many of you have heard of IVF? Just one? All right, IVF is like where if you're having problems having a kid, you know, just use a little bit of medical magic, right? They take the sperm, they take the egg, and this all happens outside of your body, and they put them together and they grow them in a dish for a little bit. But to help your numbers, you know, they don't get just one sperm or one egg, they do a bunch. And then you have a bunch of embryos. In theory, one of them, hopefully one of them works. Sometimes, very often, it's more than one. So then you have embryos. And then, again, you could do some genetic testing and say, okay, well, this particular embryo will grow up to be a child that has this disease. This particular embryo will be a healthy child. So then some people decide, not always just for disease reasons either, that they are not going to keep the particular embryos. Like, we just need the one. So yeah, you can look that up for independent work if you want to. And normally I'm not into talking about opinions, but in this case you could give an opinion. Because with the whole Roe versus Wade thing, that would be relevant, right? So if you have 10 embryos, do you keep them all, 8 embryos, right? You have just going to be octomom or not? Or at what point do you consider that okay? Those are opinions, and I'm fine with you writing about your opinion. I'm not going to give you mine, but I'm more than happy to read yours. Anyway, your book gives some examples of some genetic testing. I'm not going to ask you anything about it. You could look it up. Also, another thing for independent work to see all the different types of genetic testing. But this shows it in action, amniocentesis, right? So you can see a little baby bump, but you can't. Can you see that needle right there? He's got a needle. He's going to stick it in. He's going to get some fluid out. But they're using an ultrasound, obviously, because you don't want to poke the baby, right? You want it to be more like this, right near the baby, to get some of that fluid. Anyway, there's some other stuff they talk about too. CBS, just another way of testing. Read all about it if you want. if you want to. I'm not gonna ask you any questions about it. Again, this kind of goes back to what I was saying earlier. Once the cells are obtained, they can be screened for genetic diseases. The patient seeking genetic testing should receive counseling before and after. Obviously, I'm not gonna test you about that. And then, again, the example I gave is a little bit different than this picture, right? So I'm talking about embryos, right? I'm just talking about, cells in a petri dish, literally cells in a petri dish, not a baby in a uterus. Anyway, any questions about that? After all that talking, after 2 days, we are now just done with the first of the 3 main bullet points, right? So this is a long chapter. So let's talk about variations on Mendel's laws, because spoiler alert, most of the stuff I've told you has been slightly a lie, in lab anyway. I wouldn't say a lie, but you can all look at each other's eyes, right? You see people's eyeballs. It's not like they're either brown or blue, right? There's different shades of brown, there's different shades of blue. So everything I've told you has been an oversimplified version of how things really work. To make it even more complicated, most things aren't as simple as one versus the other, right? So that's what we're about to talk about. Let's get to reality. I'm not talking about pea plants anymore. Your book points out that Mendel's laws explain the inheritance in terms of genes passed from generation to generation according to rules of probability, right? We've already said that. That's just a reminder. So everything I've taught you is indeed valid for all sexually reproducing organisms. However, and this is the important bullet point, not for testing, but just what we're getting into, they don't explain other patterns of inheritance. And mostly, Mendel's rules can't account for patterns of inheritance. So the few things we've taught you are actually the exception, not the rule, right? So we've picked out these few true-ish examples, but now we're going to talk about some things let's talk about things the way they really happen. The first thing we're going to talk about is incomplete dominance. You need to know this. Before I tell you about incomplete dominance, let me back up and tell you this. Everything I've said so far, I just didn't call it this, has been called complete dominance, right? So with the pea flower, right? If you have that one dominant allele, you're going to be completely purple, right? Completely. That's complete dominance. There's no in between. Now, we're going to talk about things that get a little bit more complicated. Incomplete dominance. You need to know what this is for the exam. More than likely, I'm not going to give you this definition or this word. More than likely, I'll say this thing mated with this thing. This was the outcome. What kind of dominance was it? Was it incomplete dominance? Was it complete dominance? Was it some other one that I'm about to teach you? So in incomplete dominance, the F1 phenotype is a mixture of the 2 parents. So instead of it being either or, it's a mixture of both, right? So with complete dominance, right, we have purple flowers because it was either purple or white. Incomplete dominance, again, it's a mixture. So some examples. And this is how the exam would go. I'm going to say something like, I bred a purebred white flower with a purebred red flower, and some of the offspring were pink. What kind of dominance was that? Incomplete, right? Because when you mix red and pink, you get, red and white, you get pink. Or a black mouse and a white mouse, and then you get a gray mouse. Or a curly-haired dog with a straight-haired dog, and then you get the in-between wavy hair. That is incomplete dominance. For the exam, too, I'll probably make up something really wacky, something that's not even real, like a blue alien and a yellow alien mate and make a green alien, right? Because blue and yellow make green. Like, oh, yeah, it's incomplete. Anyway, here's your book example. Again, purebred red flower crossed with a purebred white flower. Those things go through meiosis and produce egg and sperm. 100% of the ones here are going to be big R. 100% of the ones there are going to be little r, because they're homozygous dominant. Homozygous for each one. So when you cross those 100% of the time, again, you're going to have a heterozygous. That part hasn't changed. The numbers haven't changed. Think about the pea plants, the purple pea plants. They were still, the F1 generation was always heterozygous. Except in this case, again, the genotype is in between, the phenotype is in between the 2 parental phenotypes. Mix them together, you get pink. Then, of course, when you cross 2 heterozygotes, you still get the same genotypic ratios. You still get 1 to 2 to 1, right? 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Except now,
the phenotypes are different because we're dealing with incomplete dominance. So now the homozygous dominant is red, the homozygous recessive is white, just like the parents, but the heterozygotes are pink. They're a mixture of the two. And again, that's a little bit more complicated. It's not that complicated, but it's a little bit more complicated than it would be in the exam. I'm probably not going to give you a bunch of numbers. I'm just going to say this thing that mated with that thing, and the offspring were a mixture of the two. Just say like, oh yeah, that's incomplete dominance. Any questions about incomplete dominance? The book is a pretty good example, a real life one, a little bit more important than flowers. Hypercholesterolemia. Right? That's a human example, so it actually involves us a little bit more than flowers. In this condition, you have dangerously high levels of cholesterol. Heterozygotes have about 2 times the normal amount of cholesterol that you should have. And homozygous recessive have about 5 times normal. And they may have heart attacks as early as the age of 2. Like, can you imagine a 2-year-old having a heart attack? I'm not gonna ask you any questions about this, it's just a good example. But here's how it works. So that particular gene codes for a protein, because remember they all do. This particular protein is on the outside of your cells, and it takes cholesterol out of your bloodstream. So if you're homozygous dominant, right, you're making it just like normal, right? You're making both of your alleles, just making copies of it. So you're pulling all the cholesterol out. If you're heterozygous, one of your alleles isn't working. So yeah, you're still making some of the protein, but not as much as you would if you had 2 alleles. So because of that, you're still pulling some cholesterol out, but not enough. So you have high cholesterol levels. So if you're homozygous recessive, both of your alleles are broke. You're not making any of the proteins, so you're not taking any cholesterol out, and you have really high cholesterol levels. Again, heart attack at the age of 2, possibly. But again, that's just an example. I'm not going to ask you about that on the exam. You just need to know incomplete dominance is when you mix them, right, and the offspring is a mixture of the two. Any questions about that? All right, the last word for attendance will be blood. And we'll just finish a few seconds early. And that's it. I'll see you guys on Wednesday, unless you want to risk it to come visit me physically in office hours. Otherwise, I'll be online.
Excuse me.
Yeah.
I remember we had an assignment that you assigned to us, and it was